Hi everybody. Today's presentation is about Mendelian genetics. Things that you need to know from this presentation include terms that are associated with genetics problems. P, or sometimes you'll see a little zero, P naught, F1, F2, dominant, recessive, homozygous, heterozygous, phenotype, and genotype. Lots of important vocab. How to derive the proper gametes when working a genetics problem, the difference between an allele and a gene, and how to read a pedigree. So let's start with who Gregor Mendel was. Gregor Mendel was an Austrian monk, and he worked in the gardens. He loved what he was doing, and he started noticing patterns within the pea plants and all the plants there really, but he started studying the pea plants specifically. Now, he was the first one to bring an experimental and quantitative approach to genetics. He started looking at these pea plants, and why would it be pea plants? Because he could control the mating. He could make sure that they were only mated with the plants that he wanted them to be mated with. And you can see in the picture here that he could cut off the pistil and use the pollen that was on that pistil to fertilize the stamen of other plants. He would use a paintbrush to do that. We still can use that same technique to cross-pollinate plants. There's lots of variety within pea plants, and they have a short generation time, meaning from the time they're fertilized to the time they're big plants and ready to be fertilized again is a short period of time. We don't have to wait a long time. And then in addition to that, pea plants can't get away. You don't have to worry about, if it's mice, about finding them and catching them. You don't have to worry about going out in a field if you're looking at sheep or goats or cattle. Pea plants, they stay put. Now, here's some of our vocabulary that you need to know. P, or parental generation, are true breeding plants. We also call these purebred. Now, what this means is that the allele that we're looking at, it has two that are the same. Now, remember that we get genes from mom and dad, and those genes we call alleles. But for our P generation, they're true breeding, meaning both of them are the same. It just happens to be they had both the same from both parents. In this case, we have purple and we have white. So this plant has a purple and a purple. This one has a white and a white. Now, F1 is our first filial generation. That means that I cross these ones, the parents, and I look and see what the offspring is. And in this case, the offspring is going to always be hybrids. And then F2, that's our second filial generation. That's when I take an F1 and cross it with an F1. So it's like crossing this with this. It would be like crossing brothers and sisters. We don't do that with people, but with plants, it's okay. And then we get our results. Now, again, when we talk about an allele, what we're talking about is a gene and all the different versions there are of that gene. So here's a piece of DNA, and here's our gene that codes for flower color. So the gene is flower color, or the trait is flower color, or the character is flower color. Has lots of names. And on one of them, I have purple, and on the other, I have white. Those are the two alleles for that gene. Now, in pea plants, Mendel found that there are 7 characters or characteristics that he could look at, and they express themselves as being either dominant or recessive. Now, dominant showed up no matter what. If it was there, I was going to see it. However, recessive can hide. If I have a dominant and a recessive together, that recessive one will hide behind that big, strong, dominant one. Okay, now these are the 7 different characters he looked at. Flower color or position, seed color and shape, pod shape and color, and stem length. Through his studies, Mendel developed these 4 principles. It's important to remember these principles. Alternate versions of genes, called alleles, cause variation in inherited characteristics among offspring. Number 2, for each character, every organism inherits 1 allele from each parent. So every organism has 2 alleles for each of these genes. Sometimes they're the same, sometimes they're different. Number 3, if 2 alleles are different, the dominant allele will be fully expressed, while the recessive allele will have no noticeable effect on the offspring's appearance. And number 4, lastly, the law of segregation. The two alleles for a character separate during gamete formation, and we'll talk about that more in a few minutes. So here's how we're going to represent our different vocabulary as we talk. Dominant, we're going to represent that as a capital letter. And recessive, we'll represent that as a lowercase letter. Okay? That helps us remember. Dominant is big, we're going to see it. Homozygous, the prefix homo means same. So this means we have two of the same alleles. Okay, homozygous. Now, in this case, it could be homozygous dominant or homozygous recessive. That's what we would call these. Now, heterozygous, hetero means different. So it's going to have two different alleles. In this case, one dominant and one recessive. Okay, this is heterozygous. So we would call this one homozygous dominant, heterozygous, heterozygous, and homozygous recessive. Next to know is phenotype versus genotype. The phenotype is what we could physically see or test for. Things like your blood type. That is a phenotype if I said you have A blood type. We can't see that though. It's not like you have a big A or a B minus on you somewhere. So we could test for that, but we consider that a physical type or a phenotype. Pheno for physical. Think of it that way. In this case, our phenotypes are either purple or white. So phenotypes are going to be words. Next is genotype. Genotype are the gene or genetic makeup of the organism. So that's going to be letters. In this case, we have a purple flower that is capital P, or homozygous dominant. Its genotype is homozygous dominant. That brings us to Punnett squares. Now, a Punnett square is a tool. It's a tool for predicting offspring from a cross. For example, I have an organism here that is capital P, or homozygous dominant with homozygous recessive. And I want to see what would the offspring be. Okay, for that, I'm going to switch cameras and I'm going to draw this out on paper because I want you to be able to see how you should do it for your testing. Okay, now, our problem said that we are crossing, make sure you can see my work here. We're going to cross capital P with lowercase p. Now, we are going to mark my capital letters just so I know which is capitals and which is lowercase because I'm not the neatest writer. Something else that I already know is that the capital is my allele for purple and the lowercase, put my mark on there, is the allele for white. I also know that purple is dominant to white. That's important. Okay, now I want to look at my possible genotypes and phenotypes. So for my possible genotypes, I know everybody gets 2. You have to have 2 alleles for every gene. And there's capitals and lower cases. So I could get a capital and a capital, or a capital and a lowercase, or a lowercase and a lowercase. Those are the only 3 ways I could mix around capitals and lowercase p's to get combinations of 2. Next, I want to know what are my possible phenotypes. In this case, that would be flower color. So I could have purple, or I could have white. And I know that if I have 2 capitals, that one is homozygous dominant. Because it's dominant, it's going to be purple. And this one is heterozygous, but because it has that capital and that dominant in there, I know it's going to be purple also. And this one is 2 lowercase, 2 recessives. When it's 2 recessives, nothing hides. I know what's there. That is white. Okay, so now I know everything that's possible. And these are my parents right here. So I want to do a cross of these parents. If I have one parent that is this and this, I need to find out what are my gametes. This would be eggs or sperm. Now, if you remember to our previous lessons, eggs and sperm are both haploid. That means they get half the information. So if I'm looking at 2 letters here, half the information means it's only going to get 1. And my choices are, well, I have this big P, or I have this big P. That's it. So let's look at our other parent. I know I can have a lowercase p, or I could get this lowercase p. So I have that and that. Now I can do my Punnett square. Move these up a little bit. There we go. I know that I'm going to have, it's going to be 2 by 2 because I had 2 possibilities here and 2 possibilities here. So I'm going to draw my box. Make sure we can see that box. There we go. And when we look, we're going to put gametes from one parent. They have to stay together. So if I'm looking at this parent, they have to stay on the same side. It doesn't really matter which side, but they have to stay together. So if I put a capital here, that means the other one has to go here. And then this one is 2 lower cases. So I'm going to put those there. All right, I have my Punnett square set up and ready to go. Now I just bring the information together. So this capital P, I'm going to put it in all of the boxes across. This capital P, I'm going to do the same thing. Then I'm going to drop these down, a lowercase, and a lowercase, a lowercase, and a lowercase. So I've completed my Punnett square. But there's still some more information that I can get from here. I can actually determine my ratios for the offspring. So I could look at my genotypic ratio, my abbreviation here for genotypic ratio. Now, so I remember, and I don't miss anything, because some of these boxes will get pretty big. I'll show you soon. For our genotypic ratio, I'm actually going to write all of our known possible genotypes. So I'm going to look back up here. My possible genotypes, we had homozygous dominant, heterozygous, and homozygous recessive. So I'll go back. And I know it's this, this, or this. Then I just go through my boxes and see how many of each I have. Now, if you're doing a larger square, you may want to keep track by putting marks in them or something along those lines, so you make sure you count all your boxes properly. This one's not so bad. If I look, I don't have any of these. 0. I have 4 of these, and I have 0 of these. So generally speaking, how we would write this is we would say that our genotypic ratio is 4 heterozygous. Now, if we can find our genotypic ratio, we can also find our phenotypic ratio. Now, remember, that's what they look like. So I need to find out what these would look like. And I know they could be purple or white. So I'm going to look at my offspring. I had 0 that were like this, 4 that are like this, and 0 like this. I know that this one and this one are both going to be purple. So that gives me 4 purple and 0 white. So I would generally write it, my phenotypic ratio is 4 purple. Okay? So I know that when I look at the offspring of these 2 parents, one being true breeding purple, one being true breeding white, that the offspring are all going to be purple. Okay, so this right here, this represents our F1. Our next cross now would be to cross our F1 generation. So let's do that. All right. To cross my F1, that means I'm going to cross my heterozygous individuals. Now I need to find my gametes. So let's say I look at this individual for the gametes. I know that I have haploid cells for gametes, so half this information means I'm going to have only one P. Well, I could have a capital, or I could have a lowercase. Now, in this case, it happens to be that the other parent is just the same. Now I make my Punnett square. I know, I can't draw squares. So I have a gamete that could be a capital, and then the other one is a lowercase. This one is a capital, and this one's a lowercase. Do you see why I mark my capitals? Again, my writing is not the best, so I need to know which one is a capital and which one's lowercase. I usually prefer to use a different letter. Letters like A or D or B. E's are great. So try to, if you get to pick the letter, pick one that's easy to tell. And then I just put my information back together again. Now you'll notice that even though I'm going across and then down, or however you want to do it, that I always put my capital first. Is it wrong to write it the other way? Not really, but we put the capital first because it makes it easier for us to understand what it is. So if I look at my genotypic ratio, I knew before that I could have this, this, or this. So if I look at my boxes, I have one like this, two like this, one like this. If I look at my phenotypic ratio, I had purple, and I had white, and I know that both of these give me purple, so that's 1, 2, 3. And this one gives me white, one of those. Make sure to put our marks in there. So I know that in the F2 generation, I'm always going to get a 3-to-1 ratio of the dominant phenotype to the recessive phenotype. And I'll always get a genotypic ratio of 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay? So let's try one that's a little different. Let's say we have parents, and we have these parents. Stop the video and try doing this cross on your own. Okay, let's do this cross together. Make sure you knew what you were doing there. So our gametes for our one parent are going to be a capital and a lower. For this parent, my gametes are going to be 2 lower. That means when I make my box, I have this and this. I put them together. Oops. I made a mistake right there. I went the wrong direction. There we go. My genotypic ratio is going to be two of those and 2 of those, and I don't have any of those. My phenotypic ratio is 2 purple to 2 white. You can't see that last little bit. Let me move it up. There we go. So hopefully you got these same answers. Okay, let's go ahead and go back to our presentation. Now we can do what's called a test cross. If we want to know if something is homozygous dominant or heterozygous, we can do a test cross. So if something shows the dominant trait, it might be a dominant with a dominant, or it might be a dominant with a recessive, and we don't know it because it's hiding. So we can test to see. And the way to do that is to cross that individual with what is the known homozygous recessive. And if we do that, we know that any recessives are going to come back out just like they did in our F2 generation. So here in these crosses, we can see that we took the individual, and if it was homozygous dominant and we crossed it with the homozygous recessive, we'd get all purple flowers. If it is heterozygous and we cross it with the homozygous recessive, we're going to get half and half. So this tells us if I get half and half when I cross it, then I know the parent had to be heterozygous. If all the offspring turn out the dominant, then I know that the parent was true breeding or homozygous dominant. Okay, now test cross are something they very well might ask you about on the AP exam. That brings us to the law of segregation. Now, the law of segregation we talked about a little bit during our gamete formation. Okay, we talked about independent assortment. This goes along with that. So, law of segregation is saying that the alleles, because they're on the 2 different pieces of DNA, they're going to be separated when we make our gametes. So if a parent has both of these, when it makes gametes, only one goes into each gamete. But we knew that. That's easy. Now, I want you to think about segregation in terms of our statistics. So if we are creating eggs and we know that it's heterozygous, that means that if I had a coin, heads and tails, just like big R and little r, and I flip that coin, about half the time I'm going to get big R and half the time I'll get a little r. Same thing for the sperm. So when I segregate them, I get this kind of ratio for our gametes, and that means that our offspring are going to look like this. So every time I cross 2 heterozygotes, okay, I'm going to get this ratio. But if you look, these here, they're the same. It's just which one's on top. But you know what? With genes, it doesn't really matter. So it's a big R with a little r. And this is a big R with a little r. So it's 1/4, 1/4, 1/4, 1/4. If I add these together, I'll get 1/4 homozygous dominant, 1/2 heterozygous, and 1/4 homozygous recessive. Okay, remember we talked about the law of independent assortment. Each pair of alleles segregates or separates independently of what is on the other chromosomes during gamete formation. So as long as they're on separate chromosomes. We're going to talk about what happens when they're not another time. So let's say that color and shape are on 2 separate chromosomes for our seeds. That means that it doesn't matter which goes where because they're going to go separately. Okay, the color and the shape. It doesn't matter if the shape is round or wrinkled or if the color is green or yellow. When the gametes are made, they're moved separately. Little more vocabulary. A monohybrid cross is when we study one character or one trait. A dihybrid cross is when we study 2 characters or 2 traits. So in this first one here, you can see we're studying just one trait, whatever this T happens to represent, but we're just studying T. Here, in our dihybrid cross, di meaning 2, we're studying 2 traits. So we have round or wrinkled seeds, yellow and green seeds. Studying 2 traits means we have more possible gametes that could be made, which means more possible crosses. So let's take a look at how to do a dihybrid cross. We're going to go back to the other camera. Okay, for dihybrids, we're a little more complicated. All right, so let's say we're talking about the pea seeds. We could look at the color, and we can also look at shape. Now, first thing is to understand them separately. So for color, we have yellow and we have green. For shape, we have round and we have wrinkled. Let me move that over a little bit so you can see everything. Oh, that's better. Round and wrinkled. Now I'm going to tell you that our yellow is dominant. Okay, yellow is dominant. So let's make that a capital Y. And green is recessive, so we'll make that a lowercase y. And for our shape, round is dominant, so let's make that a capital R. And wrinkled is recessive, little r. Now that tells us that big Y is dominant. Let's try that again. Dominant to our little y. Round is dominant to wrinkled. Okay, so if I look at these separately, let's just talk about color. I know I can have yellow or green. And then we talk about shape, round and wrinkled. But I could have something that is yellow and round, or it could be yellow and wrinkled, or green and round and green and wrinkled. See how it gets a little more complicated when we start adding in different traits? Now that's just our different phenotypes. So when I have 2 of these that each have 2 traits, that means I can have 4 different genotypes, 2 times 2. So for my possible genotypes, I have, and I'm going to use basically distribution. So if this is one area, this is the other, I'm going to distribute yellow with my round and wrinkled, so I can have yellow round or yellow wrinkled. But then I can move to green, and I can have green, round, or green wrinkled. And those are my possible genotypes. Okay, let's look at phenotypes. I know that when I'm looking here at color, I can have capital and capital, capital and lower, or lower, lower. And when I'm looking at shape, I can have this, this, or this. But when I mix them together, oh boy, does it get more complicated. So I'm going to start with my capital Y's and distribute them through my R's. So capital Y with big R, big R. Capital I with big R, little r. Capital Y with little r, little r. Then I look at the next one. Capital Y, lower y with big R, capital Y, lower y, big R, little r. Capital Y, lower y, little r, little r. But I'm still not done. Okay, now move on to the last set of Y's. Lower, lower with big, big. Lower, whoops, can't skip ahead. Lower, lower with big, little. Lower, lower with lower, lower. It's kind of crazy, right? I have 3 possibilities here times 3 possibilities here means there are 9 different ones. 1, 2, 3, 4, 5, 6, 7, 8, 9. I found them all. Now when I do crosses, I have to be able to see what's in there. So now we have to look at the possibility of the gametes being produced. Okay. So let's say we have a parent that is homozygous dominant for both traits and another parent that's homozygous recessive for both traits. So one parent is like this, and the other is like this. So let's look at this first one and look at gametes. Okay, I have 4 letters here. Gametes are haploid. Gametes get half the information. If I have 4 here, they get 2. That means they get 1 of the Y's because they get half, and 1 of the R's because they get half. So we're going to distribute. Big Y, big R, big Y, big R, big Y, big R, big Y, big R. Our other parent is like this. I distribute those. Now this means that my Punnett square, I have 4 gametes for here and 4 gametes by here. That means I would be 4 by 4. That's a 16 box cross. But with this particular one, we can cheat a little bit. They're all the same. And these are all the same. So that means all the babies are going to be the same. So that means this gamete crossed with this gamete. I put my letters back together. All the offspring are going to be like this. Now, if we look back, we can, there's our genotype for all of them, but we can also figure out our phenotype. All of them are going to be yellow and round. Okay, so our next step would be to cross 2 parents that are heterozygous for both traits. Are you ready for that? So let's change our paper here. Okay. So if we have a parent that is big Y, big R, little r, and we're going to cross that with big Y, big R, little r. Things are going to get a little more complicated. Okay, so let's look at the possible gametes for the parent. So I have big Y, big R, little r. And to get our gametes, we're going to distribute. So my gametes are going to be big Y with big R, big Y with little r, little y with big R, or little y with little r. Now for our other parent, they're the same. Whoop. Big Y, big R, little r. Our gametes will be the same. Then we have to do our cross. Now each of those gametes is different from the others, so we have to do a full 16 by 16 cross. Make sure you can see that whole box. There we go. Now I take one parent and I put all their gametes on one side. Then I take the other parent and put all their gametes across the top. Now I put them into the boxes and put the letters back together. There we go. That takes a little bit of time. After that, we can do our genotypic and phenotypic ratios. Okay, so we know what our 9 different possible genotypes are. And we then go back into our boxes and count them. So we look for how many are big Y, big R, big R. We go up to here and we count. And then we start putting numbers next to all of these. When you count, if you do it right, you're going to get this. Nope, I did that wrong. This one's wrong. There we go. 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. That same ratio happens every time in a dihybrid cross. Now, if we look, we had yellow round. Let's see if I can get them in there. Yellow wrinkled, green round, and green wrinkled. So then we count and we look. This one, definitely yellow, definitely round. This one, yellow, round, yellow, wrinkled. Okay, so you need to count, and I put little hash marks by them. But I happen to know in a dihybrid cross, they always come out 9 to 3 to 3 to 1. And think about that. Yellow is dominant. Wouldn't I see that the most? And round is dominant. Wouldn't I see that the most? So together, round and yellow, that would be the most of all of them. And then yellow wrinkled and green and round. One's dominant, but the other's recessive. And then the last one, green and wrinkled, recessive and recessive. There are going to be very few of those. Now, sometimes the biggest part, the hardest part, is figuring out the gametes. It doesn't matter what our trait is. We can have any different combination of letters. Let's say I have this, 5 different traits, and I need to find what the gametes are. I distribute. That's all I do. I distribute. I can put my big A, the big B, a big C, a big D, or a big E. Or I can do the little e. I can do a big B, big C, a little d with the big E, with a little e. And you just keep going through and finding all your possibilities, changing one set at a time and working your way through. There are 2 possibilities here, 2 here, 2 here, 2 here, 2 here. 2 times 2 is 4, times 2, 8, times 2, 16, times 2, 32. There are 32 different possibilities for gametes. Now let's change this up a little bit. What if I had this? How many different gametes can I get? Well, I have 2, 1, because these are the same, 2. 2 times 1 is 2 times 2. That means I'm going to have 4 different types of gametes. At this point, from here on out, I wouldn't make you make any boxes. If you can do a monohybrid box, you can do everything else you need to do with math. Okay, that's it for our Mendelian genetics. Next is our non-Mendelian, things that break these rules. And then we're going to talk about some more math, some probability, and figuring out what predictions we can make. Bye, everybody.